Okay, everyone, you can have your attention, please. I have the pleasure of introducing Dr. Mikhail Tikhonov, Assistant Professor of Physics here at WashU. Dr. Tikhonov's work lies at the intersection of physics and biology, with a focus on microbial communities. His lab combines experimental microbiology, bioinformatics, classical ecology, and statistical physics to study microbes. To borrow a little bit from Dr. Tikhonov's website, which I liked. See if you agree. The challenge of understanding microbial communities is not just experimental and technological; it is also theoretical. Microbiology and microbiome research are revolutionizing biology, making us question some of the most basic concepts, such as species, fitness, and even organism. What if our macroscopic intuition about ecology and evolution is simply wrong at the scale of microbial life? Dr. Tikhonov's professional history: he's assistant professor starting in 2018. 2017 to 18 was a postdoctoral fellow at Stanford University, and from 2014 to 2017, he was a postdoctoral fellow at Harvard. We're talking today about emerging simplicity in microbial communities. As usual, there will be a reception afterwards outside Bear Hall with refreshments and an opportunity to talk more with Dr. Tikhonov. So with that, please join me in welcoming Mikhail.
Thank you very much. This talk is meant to be much more practical than philosophical. Also, this is not the audience where I would claim that my work involves a lot of classical ecology. That works more with physicists, but you be the judge. Anyway, yes, so thank you very much for having me. I've been enjoying the series, and it's a pleasure to be giving one of these. I am indeed local here in physics, and I think about microbial communities and what's the relationship between physics and microbial ecology. That's what I hope to discuss today. Okay, very good. Yes, so this being the Living Earth Collaborative, all of us think a lot about biodiversity, and my research also involves thinking about biodiversity, but at the primarily at the microbial scale. So there's a study of microbial ecosystems. They are everywhere and incredibly complex. Here it's a broad audience, so I don't know if you're necessarily thinking about microbes on a daily basis. Some tidbits that people often put in the introductions to their talks is things like, you know, the volume of space that you think of as you is not actually you. It's mostly microbes. There are, 10 times as many bacterial cells in there than your own. Very important for not only for disease, but also for health. And that is somehow weirdly human-centric, but microbes are everywhere. They are in the ocean, they're in the soil. They shape some of the largest scale processes on the planet. So not just our health, but also the health of our planet. And wherever you look, they're incredibly diverse and complex. There's hundreds of species all coexisting, all interacting. And how are we ever going to predict anything? That's kind of the question. And as long as, I would say, kind of as old as ecology, theoretical ecology, there's this hope that despite this complexity, some properties might actually be predictable of these complex systems. That the search for such properties has a very long history. Something that is a more recent idea is that perhaps some properties might even be predictable not despite diversity, but maybe even because of diversity. And that is kind of an interesting idea that connects in particular to this branch of physics that I was trained in, statistical physics. So statistical physics is an area that studies emergent laws in complex systems. With a kind of a canonical example is, for example, the gas in this room, right? There are trillions and trillions and trillions of molecules. They all interact. Trying to understand this on the microscopic level would be absolutely impossible. And yet, you know, if you think about some macroscopic properties like temperature, volume, pressure, there are some regularities that link those properties that you can predict despite not knowing all the microscopic details. And it's been a tantalizing hope for a long time. Could something like this also maybe apply to this interface with ecology, where there's also some parameter that is large, such as large number of interacting species, for example? Are there any regularities that emerge from that? Now, ecosystems are incredibly more complex than the gas in this room. All molecules are the same. All species are different, right? So this is kind of a aspirational analogy, but it's an active interface area, and we're doing what we can. So, like I said, it's a long history, but like especially this idea somehow is very appealing if it's true, right? If something could be predictable because of diversity, wouldn't that be amazing? That would maybe open new avenues for thinking about natural ecosystems. Because one of the dominant tools we have is to bring things in the lab and into simplified conditions, maybe simpler ecosystems. That's what people do with microbes specifically, right? And maybe if there are some strategies that could work, not despite diversity, but because of it, right? Maybe we could go and try and understand things directly in their natural context. And maybe that would be so cool. Okay, that would be amazing. How well are we doing on that? Not great. So the search for such emergent regularities, I will put it under kind of two umbrella categories. One is, I'll call it emergent reproducibility, which is, of course, way older than anything even microbial. So classic work from the 70s, people thinking about like the ratio of, you know, kinds of predators to kinds of prey in across food webs is, you know, reported roughly constant, or roughly stable ratios of some functional groups across different ecosystems, right? These kinds of ideas of what is the same as details change. And in microbial world, there's a lot of that as well. So like one example, if I look at some, this is from synthetic data from lab experiments, I assemble some ecosystems. And if I look closely at the strain level, they're all different. But if you coarsen them a little bit, maybe at the level of taxonomic families, oh, it's more reproducible. And big literature in that often has this name to it, functional convergence. Often the words people say is, you know, function is more predictable than composition. I have to be careful here. I had this conversation with Joan yesterday, who emphasized that I need to be careful with words, which is absolutely true. So in this talk, when I'll be, whenever I use the word function, it is, and I'll try not to, but when I do, it is by contrast with composition. So I mean any property that's not just abundance of some species. I mean a process rate. I mean, you know, how resistant, how protective is this community against an invasion? How resistant is it to somebody, to invasion, right? Or stability also, right? Any property that's not composition. That's what I mean by the word function, without implying any teleology in the background. Okay, so there's a lot of that. Another example, you start with communities, they're kind of all different, and then they converge over time to something more predictable. Okay, there's a problem with this emergent regularities. One is, you know, there's a lot of debate to what extent these examples actually generalize, or if it's wishful thinking, how reproducible is reproducible. And like I cite a recent paper that was actually about this same observation. Well, it's true if you assemble communities in a simple medium, but not in a more complicated medium. So, you know, a lot to discuss. But maybe more importantly, even if it's true, I claim we want more in ecology. So as an illustration, let's say consider a statement that if the soil community is, I don't know, complex enough, diverse enough, the rate at which it will fix nitrogen will be roughly X per gram biomass. That blanket statement will probably be somewhat naive, right? That's one issue. But imagine even that it is true. That leaves some curious questions unanswered, which is what do we do to increase it, to decrease it, right? How useful is it that future examples will look a lot like previous ones? That's nice, it tells us something, but we want more if we want to predict something about the behavior of these systems. And so accordingly, a lot of physicists actually are excited about reproducibility. Ecologists have been looking for a lot of emergent correlations. Oh, sorry, so my reference block disappeared. Okay, so relations like, diversity function relationships, diversity stability, like productivity of a community is a function of diversity. So there's a large literature on that. So this is, I just took from paper Alicine and Tang, 2012, I think. Like, if you have more predator-prey-like interactions in an ecosystem, maybe it's more stable, right? These kinds of predictions. So this is nice because it is explicitly focusing on something that, at least in principle, has some predictive power implied there, right? It is closer to making some prediction. But the evidence for these things is often conflicting. There's a lot of debates. People report correlation positive, negative, or meh, right? So suggesting that maybe also this kind of expectation is maybe a little too simple. And overall, this can mean one of two things. Either this whole idea of emergent simplicity is sort of theoretical aspiration that is very nice, but doesn't survive the complexity of the real world. That would be very disappointing, not just for me, but I think for many of us, because in some sense, this has been the role of theory in ecology, right? To propose such relationships that you can then go and test, or to organize our experimental data in some way to prove a hypothesis, disprove it. But it could also mean that there are any emergent regularities that are true, maybe don't necessarily reduce to something simple like a correlation or a universally observed correlation, right? Because a lot of these, even if it's positive or negative, well, in some contexts it's positive, in other contexts it's negative, right? It's like a more dimensional concept. Okay, so that's kind of the setup. And my proposal for today is to try and move beyond correlations to this idea of predictive power of simple models. And can we quantify that, and can we see that in data? How do we proceed about that? And let me start by giving you a couple examples where I'll claim a simple model or simple description was successful at describing something complex, capturing behavior of a complex ecosystem. And this is all in microbial context now. So here's one. This is a cartoon representing an industrial bioreactor. It's treating organic waste that kind of flows in. Inside, there are microbes that digest that mostly into, and methane comes out the top. So if you ask who's in there, it is incredibly complicated. There's like over 1,000 strains of bacteria, 100 strains of archaea, right? Extremely complex. There are hundreds of relevant metabolites in there. But turns out that if you want to predict how much methane it's producing, which is in some sense the goal, turns out that you can ignore all that diversity to a first approximation, coarse grain it into 4 classes by metabolism. Instead of hundreds of metabolites, you can think about like 6 main categories, and it is reasonably predictive. People who actually run these reactors industrially, they have much more complicated models that take into account the shape of the vat and all sorts of things. But like this does remarkably well, given how complex the system is. There is an interesting theoretical puzzle here that I'll kind of put in as a plug, because you might wonder, how can this actually work? how can a coarse graining like this be predictive if it's grouping diversity that we know is functionally relevant? So we know that in an ecosystem, in a microbial ecosystem, the functional repertoire is shaped at the level of strains. There's lots of papers showing that. And yet somehow here, we take all this diversity, ignore most of it, and still get prediction right. So that's a question that I'm very interested in. We have some, theory land papers on that, but we're making progress. But I'm just putting that as a plug. I'm not talking about this today. For today, I'm just using this example to say that there are examples when a surprisingly simple description is predictive. So in this case, it's a simple description. I took a very complicated system and I coarse grained it to just 4 classes. Here's another example. This is our recent work where we looked at how predictive can be simple models. So we looked at data sets in the literature that are of this kind, where these are all synthetic lab experiments, where imagine you have some set of species that you mix in some combinations, and for each, you stabilize them in some way, and then you measure some property that you care about, this property Y. And now the question is the sort of structure to function mapping, it's called, right? So can we predict Y given the composition? And we asked, can we do this? Let's do something really dumb. So let's try and predict it using a model that is just a simple regression. In fact, it's a regression. It's even worse because this here is just the presence or absence of a species. I'm not even putting in abundance. I just said, did I put it in originally or did I not put it in? How well does this do? Yes, I was warned this would shift. Okay. So this would be a very dumb thing to do, except it works. So we looked at 6 examples, 6 data sets with different microbes, different properties, kind of gut, derived species, soil-derived species. And across these examples, we get our square of 0.8-ish. So it works surprisingly well. Now, what do I mean by surprisingly well? Like, should I be impressed by our square of 0.8? How do I know? The comparison here, so in some of these cases, people actually did a much more careful mechanistic modeling of this community, in particular, like in this paper. Way more complex model, and we do just as well. That is not necessarily a comment on how well regressions work. This is maybe more of a comment on how hard it is to actually fit a logical Terra mechanistic model to some microbial ecosystem. But be that as it may, I won't go into details of this. This paper is out. I'm just using this as another example where surprisingly simple models can also be reasonably predictive. So inspired by these examples, I want to ask this question that I sort of started with, which is there evidence for simplicity actually facilitated by diversity? Because that was this exciting idea from my slide 2. And these examples don't tell you that. And I would say that previous literature does not actually resolve this question. So if you think back, the bioreactor, that was very high diversity, right? 1,000+ species, something was predictable. The examples I just showed you were between like 15 and 25. And nobody tried varying systematically community diversity for this question: how predictive are simple models in that context? So what do we need to answer this? We need some quantitative framework for characterizing the simplicity of prediction that we could use to compare across diversity, and that would actually be measurable. And I want to emphasize, like, what's the sort of hard part of this question? If you have a hypothesis, like, I hypothesize these two things are correlated, I can go and measure that correlation, and I can observe if it's stronger or weaker. I can ask if it's statistically significant, right? I know how to do that. But how do you ask this question? Here's the data set. How predictive are simple models without it seems like I'm not going in with a hypothesis to test. It's just how predictive are simple models in this data. How would you do that? And how do you do it in some not ad hoc way, but in some systematic way to quantify this? So this is the work of fantastic graduate student, former graduate student, Jake Moran. He is now postdoc at Michigan as a pioneer fellow, sadly, no longer here. Okay, so let's make this slightly less philosophical and more concrete. Here's an experimental setup that I want you to imagine for this discussion. Let's say that I have a fridge with 50 of my favorite strains that I will use for the experiments. And I can take them and mix them a few strains at a time. And I'll call this low diversity data. And I can also mix them, 20 at a time. And that will be my high diversity data set. And for each community, again, I stabilize them in some way, maybe passage a few times. There's some protocol that I adopt, and then I measure some property that I decided I'm interested in. Now, what would be here this emergent simplicity that I'm looking for? The first variance, this emergent reproducibility, you might observe that in the low diversity case, this property is kind of all over the place, right? But as you go to these more complex communities, somehow the distribution becomes tighter, right? And that is this emergent reproducibility example. And that's the thing that I claim we want more. That's not enough. So the next thing is the emergent correlation would be something I plot maybe y against some compositional feature, again, not correlated, and then you sort of observe this correlation. And beyond that, some maybe higher dimensional relationships that I'm trying to generalize this to. This is what we're trying to do here. Make sense? Cool. All right, so here's a framework for interrogating emergent simplicity. The goal, I remind you, is to quantify the statement, whether simple models become more predictive at high diversity. I'll draw inspiration from these two examples that I showed you. So in the case of the bioreactor, what was the simple part? I had thousands of strains and I coarse grained them into just 4 classes and it was predicted. So here are my strains from the fridge. And my central object will be this notion of a coarsening. I group them in some way. In this case, maybe in 3 classes, and I will call the combined until this. And now that I have this coarsened description, I will fit, let's say, a regression model to try and predict the property of interest y. And I'm using regression because inspired by this other example where things worked. But here I'll be using actual abundances and not just presence absence. So why we're doing this, the goal is to, quantify whether simple models become more predictive. So this is my simple model. Instead of looking at this whole glory of complexity, I coarsened my data by grouping, combining taxa into coarser groups, and then I fit a regression to those coarsened buttons. Now, there might be various ways you could try and guess what's a sensible way of grouping your taxa. Maybe this could be taxonomically, right? Or maybe by some annotation of some gene or what you know about what these taxa do or something like that, right? So there could be candidate coarsenings that your biological intuition might suggest. Here, these all provide an example of coarsenings, but I'll be agnostic. I'll allow any coarsening, and I'll let the sort of algorithm churn through them and select what it likes. So given that, what I'd like to do now is I'd like to be able to make plots like these. And of course, importantly, I want to be able to make plots like these from data, right? That's where I'm going. So we'll see that in a second. So I'd like to ask, if I'm trying to predict property y, I'd like to be able to make some diagrams where on this axis is how complex is my model, and on this axis is how predictive it is. And if I knew how to do that, I would then look at what this looks like for my low diversity data, high diversity data, and compare. That's the goal. Okay? By the way, also, usually speak to audiences of, physicists or some quantitative biology but like, please interrupt me if I'm using some jargon or something, or for whatever reason. I'm not saying anything profound, so I should be able to explain it clearly. Okay, so that's the goal. And the question is, what do we put on the axes exactly? And the beauty of this setup is that once I set up the problem for you this way, there's actually essentially only one natural thing to put on both these axes. Like at this point, this is already clear how to proceed. So model complexity. How complex is my description of the system, right? I can tell you every single detail and ask how well can you do within your model class, or I can, here's a kilogram of biomass I dug up from the soil, predict this property. What do you think this process rate is? Good luck, right? So that is the 1 kilogram of biomass that's here. And if I give you abundances of every single strain, that is here. So essentially, like the number of classes that I coarse-grained into is a sensible thing to put on the axis. This is essentially what we do. We do a little twist. So what we put there is what's called the entropy of this partitioning. It's essentially the same thing as the number of classes. It just correctly takes into account that if my classes that I've broken into are not the same size, they shouldn't all count equally, right? But there is a sort of field where everybody would agree that what we're putting on here is the only sensible thing to put there. So we did not invent that. So that's the complexity description quantified by this entropy or information content. There's some formula. Formula does not look intuitive, but it's essentially a number of classes. And the y-axis is even simpler because what I want is predictive power. Once I train this model, how predictive is it? Well, let's just look at prediction error and plot that, right? So those are the axes. Yes?
Clarifying question. Is the number of taxa going into this the number that went into the beaker or bioreactor at the beginning or the end? Or
oh, so this is my complexity of the description, not of the community.
But in
right?
the communities themselves, we're thinking about how many taxa are involved.
Yeah, so
more diverse.
So I have a fixed library of species, let's say 50 in the fridge, right? And then I decide maybe let me coarse grain them by taxonomic family. So instead of 50 species, I could describe with just 5 families. So that would be the number that I would put on here. And then I would ask, if I'm using some model that ignores the division into species and only uses information of abundances of families, how much error is it making when I'm trying to predict this property in ecosystems that are actually diverse, high diversity, like maybe 20 species mixed at a time? Or if I look at 2 species at a time. So that is, the complexity of the community. I will apply this to simple communities or to complex communities, and each will have a plot. But one plot here, this axis is the complexity of my description.
No, I understand that. I just wondered, like, when you say of 20 species put into the community, you're talking about the number that entered, not necessarily the number that
survived. Oh yeah. So this when I was talking about presence absence, that is a very important question, and that's why now I'm not talking about presence absence. I'm talking about the actual abundances.
Measured at the end.
Measure at the end. Yeah. So measure at the moment when I'm measuring my functional property. Yes. Thank you very much for the question. Okay, we're good. Okay, so this will be my main kind of object here that I claim we can actually make for data, and I claim it contains a lot of information. So let's understand what are our expectations. What do we expect it to look like? So in general, it will look something like this. So let me first look at this point here. I call it point Z for zero information. This is when I gave you a bucket and said, here's a kilogram of biomass, predict property Y. Or actually, yeah, so I told you exactly nothing about the actual community that I asked you to predict something about. How well are you going to do? Well, all you know is, in general, this property is somewhere between this and that, right? So if I told you nothing, your best bet is to predict the mean. And then the error you're making is how wide this distribution is. So if this point is high error, that corresponds to this scenario. If it's low error, if it's a very reproducible property, it corresponds to this scenario. And I'm walking you through this to show how where this point is, that encodes this, whether I observe emergent reproducibility or not, but our framework generalizes that. There's way more information here. So now that I actually give you some information about composition, how much better can you predict? So there'll be a lot of points there because there's a point for every possible coarse graining I might consider. And there's an astronomical number of those. And some of them might be biologically inspired and others may look silly, right? All of those are points in there. But there is some boundary to this cloud. This boundary is called the Pareto front. This is like your best bang for the buck. If you give yourself a budget of some complexity for your model, right? I'm willing to describe my ecosystem with up to 10 classes, right? You can always choose them in a silly way to do terribly, but what is the smartest way you can pick? What's the best prediction power you can gain that by, right? And that traces out this curve, and that's the important curve for my purposes. And importantly, in general, we would expect that towards the end, this is what my cartoon is meant to represent, towards the end, this curve will generally actually go up because, and that's a known phenomenon, that's overfitting. So of course, when we evaluate prediction error, we do out of sample, right? I hold out some data, I predict, and if I have, 100 parameters in my model and only 50 data points, that's a terrible idea. So given the data that you have, beyond certain complexity, don't go there, right? So we know that, and somewhere in here is our golden spot. That's kind of the best good enough model that we can construct within the model class of models I'm considering, in this case, regressions. Make sense? Cool. So my question of emergent simplicity, what would it look like in this language? The sort of golden hope would be to observe something like this. So what is this showing? As I apply this framework to data on low diversity communities and then high diversity communities, if I see my point shift in this direction, what does that mean? That means that as diversity increases, simpler and simpler models are more and more predictive, right? You have less and less prediction error. That would be amazing. Okay, one comment here, which I kind of partially alluded to, but I want to emphasize because that's important. What's on the axis here is complexity of the description, not complexity of the community. So in all these cases, I have my fixed fridge of 50 strings. And for example, this point here, the microscopic description, is I'm fitting a regression model with 50 parameters, right? Same model, but I'm asking, is it more predictive? Does it make more errors when it's describing communities like this or communities like that? And that is the question. Yes?
Is it more important that the high diversity Pareto front decreases in complexity or decreases in error?
Very
Or
good.
does it have to?
I would say either would be great. I mean, at this point, you should be asking yourself, that would be amazing, yes, but what are the chances that we would ever see anything like this, right? I would be happy with either. I would say that what we have evidence for is it going down. But either of those I would call emergent simplicity. And in fact, that's the reason why, like, this sort of language is interesting, because this kind of diagram encodes a lot of these, like, instead of just using some buzzword, we can now like distinguish different kinds of simplicity. Does it get, does it go down or does it go left? Excellent question. Okay, so this would be great, but what are the chances of ever seeing this? So let's go find ourselves some data. We have a number of requirements that a data set needs to satisfy for this to be applicable, because we need some data set where people assayed communities of sufficiently varying diversity for this to be a reasonable thing to imagine on the x-axis, right? And we need something with a large enough number of data points over number of strings in the pool. The reason I'm using something as simple as a linear regression is because I sort of foresee trying to apply it to actual data. which will be limited. And so training something more complex in the regression seems almost hopeless. But even there, right, there's really cool data from, for example, grassland experiments. So we wanted to do this not just on microbes, but also, how does it work for classical ecology, for grasses? There's a real cool data set where people actually did this combinatorial design, combining grass and different combinations and plots. This went over something like a decade or more, incredible amount of work. And we cannot use any of this because they had 100 plots, but these are combinations of 50 grasses. And they actually did different, a few grasses at a time, more grasses at a time, precisely to check this kind of diversity, productivity relationships, right? But we cannot use it here because we cannot constrain a model with 100 parameters with only 50 data. So with these constraints, we found basically 2 data sets that we could use. And I want to emphasize these are not 2 data sets on which this worked. They're the only 2 where we could find to ask this question. Right? So one of them is this Clark et al. Absolute tour de force of combining up to 25 strains and measuring butyrate production, acetate, lactate, succinate. There's this belief that butyrate is a very important metabolite for the gut communities. So they sort of, that was the motivation for this work, 25 gut-derived species that they combine in various ways. And the number of strains that they put, right? We can actually do this. These are our low diversity communities, and these will be our high diversity communities. We use 15 and higher as our high diversity. And there's one other data set that's only measured one thing, but a different, combining 14 strains up to 14 at a time, and one of them was labeled with fluorescently, so the abundance of that strain was the property they measured. Make sense? Cool. So what we're looking for is signature like this, and this is actual data, and 4 out of 5 cases actually show, we were kind of shocked when we saw this. I had my students check absolutely everything. So we did a bunch of tests on this, like randomizing labels, randomizing species compositions, perturbing slightly how your algorithm searching for the best grouping, perturbing that grouping, subsampling your data. We are sure about everything that's on the slide. So this is the curve, the Pareto front, that for the high diversity communities, there are error bars on there because we do this, you hold out some data, right? So over different random splits of what you hold out, you get slightly different numbers. That's the error bars on the Pareto fronts. As pictured here, all of them show this pattern, but as we do our testing with subsampling and things like that, here error bars, to be conservative, I would say this is within error bars. So that's why I'm saying 4 out of 5 and not 5 out of 5. Okay, so let's kind of zoom out. I set up some fancy language for you, threw some information theoretic words at you, and said there's entropy, and we can do a thing, and it shows emergent simplicity. Now, this is not some fancy information theoretic statement. We can actually reduce it to something much more intuitive. So now that an algorithm returns this, right, we can just go and look. Here the claim is how to read this plot. This says that if I look at various models of this complexity, right, the best model that, as it fits the low diversity data, can only explain 30% of the variance. But for the high diversity data set, there exists a partitioning that explains, here's the 70%, right? So what is that model, right? Here, there is some very simple model that supposedly explains it. Well, let me just make a scatter plot. This is butyrate production against this compositional feature that's this identified. This is in the high diversity case. For the low diversity data, it's not the same feature. The best feature that correlates the most, the scatter plot looks like this. So it is very obvious once you know where you're looking, right? But you needed some algorithm to identify that for you. And because there's a large number of possible groupings, that's why we needed to be very careful with multiple hypothesis testing, randomization tests, right? And we did all our homework. So now the question is, okay, very good. This is real. I was very excited. Why do we see this, right? Why is it so much more predictive here? And actually, so for this specific case, with specifically butyrate, the original paper describes, and we talk also to the lead author on that, there are hypotheses I can sort of throw at you that might be suggestive. So for example, actually in these communities, there are 5 strains that are capable of producing butyrate. And at low diversity communities, all 5 contribute. But it so happens that when you go to higher diversity, you will also include another class of strains that modify the pH of the medium in a way that 4 of them do not like. And so in the high diversity communities, typically only 1 of them actually contributes, and its abundance is very nicely correlated with the overall production. And that is, in this case, the simple compositional feature is just the abundance of that one butyrate producer. In all the other cases, there's not like some one species that contributes. This is just one example I'm showing because they have a story to tell to possibly explain how that may happen. In fact, the authors also, they even actually hypothesize that that strain senses the diversity of the community, like it actually changes its metabolism as a function of whether they are in the presence of these other strains, which is very cool. They're testing that, right? I don't have more to say on that. There's like another hypothesis. What I want to emphasize though is that this is for this case. I don't have a mechanistic story to propose for the others that would require more, mechanistic work, more data. But those would be 5 different mechanistic stories. And the fact that in all 5 cases, yet you see this behavior, maybe is exciting because maybe there's something more general. That's my hope as a physicist. So what could be something more general here? We have a sort of candidate mechanism that we're trying to substantiate in this particular case as much as we can. That's why this is not yet out, because we're trying to see if we can push that a little bit further. And the idea is to relate this to incomplete functional convergence. And here's what I mean. This is now cartoons. So imagine that I take some low diversity communities and I scatter plot some 2 properties. Now, I'm not even talking about composition versus some property Y, just 2 properties, which is actually how, like in this case, this data set measured 4 functional properties: butyrate, acetate, succinate, right? That's how we're actually trying to relate this. But this slide will be a cartoon. Let's say you do the scatter plot, it's kind of all over the place. There is an idea that has been discussed how diversity can lead, I mentioned this at the beginning, to some form of functional convergence where some things, if you sample from a larger pool, become more reproducible, right? And if you observe this functional convergence in all dimensions, then what you get is the scenario of emergent reproducibility, right? All observables are very tightly distributed. That's your reproducibility. But now imagine what is arguably a more general case. What if it's not in all directions, right? Imagine there's functional convergence in some directions, but not others, or in some more stronger than in others. In this simple 2-dimensional case, if there's some direction that shows this convergence, that would lead to an emergent correlation. And in some general case of many observables and, inhomogeneous functional convergence, you might imagine that this will therefore give you some higher dimensional relationships. In this case, I said that, let's say, all dimensions converge except 1. So this kind of, at low diversity is some big cloud, and high diversity converges into some banana. And there's this banana coordinate that is secretly, you measured 4 things, but secretly there's a property of the community that actually it's lower dimension. That's a lot of the structures that you could get are actually forming that. Yes, there's a question.
I'm understanding how you're suggesting convergence here, but versus, let's say, engineered mixed models, when your model fails, where there is incomplete convergence, you see the errors increasing much more. So in here, would your emergent correlation would suggest that your predicted power is going down and you have like more error rates? Or how are those two things related?
In this case, my predictive, what I previously discussed, so there's this sort of little twist that happened, because everything that happened before was about composition to functional property mapping. And I was training regressions to predict this based on this, right? And so in this case, if that happens, well, if this is a tighter cloud, then you have lower error prediction. But the switch that happened is that people describe functional convergence in the space of functions of like, maybe, okay, I don't know if that actually helps. We can discuss after this, just to make sure that I understand also what you mean by the linear mixed models. Okay, all this is to say that in this one data set where they actually measured 4 things together, we can directly look at this, and we have evidence that actually high diversity functional data does collapse into a banana, and the banana coordinate there is actually related to the pH, which actually makes a lot of sense given what they described in the interactions of these communities members that are pH mediated. So that is a final part of the story that we're still working to like actually nail down, but that will provide a little bit more of a mechanistic point here. Okay, and with this, let me just summarize what I think I told you, and you can tell me if you agree. So I'm excited about these results because I see them as providing evidence for emergent simplicity as a path to predict something about the behavior of a community. And at least in these examples, we can actually, quantify this statement and show that as community diversity is increased, simple models become more predictive rather than less in this very formal sense. So what should we do now? The first obvious thing is I advertise this to you as this would be amazing if it could somehow let us access, natural ecosystem. Everything I told you was for up to like 25 strains, which is a very far cry from anything resembling natural diversity. So how would you extend it to that regime? And in particular, the computational problem that here is sort of swept under the rug, I just showed you, there's an astronomical number of possible groupings, and here's the Pareto front. All of the work was actually in how do you search that space to find a Pareto front, right? And I didn't even talk about that at all. We can do that in this case. For some, what we're doing now for a natural, soil example sample with hundreds of species would not work. So we need to be clever somehow, and that's one direction that we're exploring. And the other one is, how do we actually turn this into learning some new biology from this? Because I told you, look, it is predictive. The hope is that, well, why is it predictive? Now that we have found something that's predictive, rationalizing the success might tell you something about the mechanism, right? That's kind of the hope. Assuming that is actually causal, not just correlative, right? That's so all of these are remain to be addressed. So how are we going to get there? This is my meant to be a segue, but it's like not really. Here's another thing I want to tell you. This is just one slide, which is, so this is kind of one direction of what I'm working on in my group, with sort of coarse grainability and emergent simplicity. That's what I'm excited about. And we also have a hobby, which is to work. Oh wow, these were meant to appear sequentially, not all at the same time. Sorry. Okay, so another thing we like doing is working with collaborators who have like really cool, highly quantitative data with like small error bars. And then we work with them to make those error bars even smaller and correct systematics and like get more out of this data. And I meant to sort of show you a few vignettes of, over the years, like works in development, like single molecule imaging of transcription. And, we were joking how it's, error bars, and then do you have error bars on your error bars? Like, how well do you know your errors, and why does it matter? Well, these are different examples where we actually could use this high quality of data to gain insight into the biology. So in this case, because we could actually see individual transcripts and count, see transcription in absolute units of how many transcripts per minute, right? We could actually show how across gap genes in Drosophila, like the highest rate of on is actually the same and it's limited by some physical, limit rather than just like, oh, it's regulation. You don't know anything about it. It could have been anything, right? So that's on the scale within a cell. This is actually in 16S sequencing. This is pretty old, but we're among the first waves of developing high resolution bioinformatics methods for 16S sequencing. And so if you sort of look at two sequences that are these are time traces of 16S sequences that are very close, you can do some fancy time series analysis to actually identify resolve cases where they too come from very similar strains, but physically distinct, from cases where actually these are two 16S copies on the same operon in one bug, which is kind of cool, because we often treat them as proxies for lineages, but actually one bug can carry multiple 16S copies that could be actually different. Sometimes they are more different than those that clearly behave differently ecologically. So anyway, there's some stuff like that. Worked with a collaborator who was watching very carefully cells grow under a microscope, and then they would change temperature. And the individual traces look like this. But if you look carefully at the growth rate, you can actually see that there's a peak in there. And I worked with them to identify finding error bars here to convince ourselves that this peak is real. And this peak actually tells you stuff about how the cell regulates its growth. Again, in a way that could have been regulation and therefore anything, but actually it's a general property of just coupled reaction networks, things like that. They get some biological insight, and this is some more recent thing on, correcting biases in amplification biases in fitness assays, whatever. So part of what I'm telling you this is because one of the reasons I'm here is to meet everybody, and people have cool data, and we like working with cool data. So that's my plug. Now I know what you're thinking. It's time. Is that what you're thinking? Okay. You're probably thinking that for somebody with a passion for small error bars, ecology was the bad choice of career. So that was indeed sort of a major source of unhappiness for a few years for me. But I want to advertise the work of some fantastic collaborators with whom I'm now working. This is a group in Chicago, Sepikun, with a very talented grad student who generated an incredible data set on soil microbiome, which is the cleanest you will ever see. And so we're very excited about that because this is a place where we can actually harness those error bars in a way that is reasonably standard in some fields like quantitative imaging and is totally amazing what you can do with kind of microbiome scenarios. And this is another group in MIT, Otto Cordero. Here they have we're starting to assemble some data from marine communities where we could actually test these ideas of what I described to you as this algorithm for identifying some groupings. Might this be a good way of, in some formal sense, define something that could be used as functional groups for a given purpose, right? Where my evaluation of how well the algorithm did is not whether it made sense to me, oh, I grouped these and that kind of makes sense, they're all denitrifiers, right? And this one is the odd one out, but maybe it's okay. But in some formal sense, I have a criterion for what makes a good grouping is if I can actually predict something from that. And so we're gathering data on this where you can independently validate it by perturbation experiments, because things that are presumably functionally similar should respond similarly to environmental perturbations. That's the kind of data we're trying to get. So that's the exciting future. And this is my conclusion slide. Just the bullet points, some more speculative points that I didn't get to talk to, and it's fine. And thank you, everybody. Yeah, thanks for having me.
Okay, wonderful talk. We have time for a few questions.
Thanks, that was a really great talk. I have one question is that the data that you're looking for or the ones that you're doing predictive power with seems like things that are measured in large numbers. So things that you would take a logarithm of and then you're measuring some kind of central tendency. But what about the kind of data where you are looking for rare events or looking for a particular rare, let's say when you're variant calling, you're looking for one particular mutation. Is your method, like, do you have something in plan for how the predictive power of the rare events, or does it just matter for things that can fall into a distribution with central tendency measures?
Okay, there's several things there. So, oh man, okay, in what order? So one comment is there is a general field of, for example, in machine learning, of feature selection. And you could think of a lot of what I told you as like, here's compositional data, here's some function we measured, what's the most predictive feature, right? There are general purpose, there are attempts to do something like this with an algorithm that knows nothing about what's under the hood, right? It could be biology, it could be, or like, it could be ecology, it could be some variant calling or whatever, some general purpose thing. And we're not doing that. We're trying to kind of be inspired by this idea of grouping similar tasks together in some, that's inspired by this notion of functional groups, right? And I don't know if there's an analog of that. And there are lots of other contexts where feature selection like questions arise, and we don't have anything to tell those fields, right? So it's a very distinct question. Now, the other thing that you mentioned was like something like central limit theorem, like, oh, it's because of large numbers, there's ideal self-averaging. This is actually one of the reasons, one of the motivations people have for like, why might it be true that high diversity communities are more predictable? Well, you know, it's some sort of self-averaging type argument. So I usually address that directly because I talk to physicists where their first thought is self-averaging, and I didn't. And maybe that was a mistake because maybe that's the sort of intuition that a lot of you had about like what we're trying, that a lot of what I'm showing you maybe is self-averaging. So it's self-averaging is this kind of emergent reproducibility, right? If your quantity is self-averaging, it will become tighter and tighter distributed. Here, I am purposefully trying to ignore that because I want to say that is the boring kind. I want to go beyond that. And so to do that, all these curves that I showed you, remember that the self-averaging part of this, oh my God. Okay. The variance of the distribution is this point at the y-axis. And notice how here in all curves I'm showing you, it's 1, because I normalize them to the variance of the observable precisely to ignore this simple, oh, law of large number effect. I am explicitly looking at, given that, if I give you more information about composition, how much more useful is it for you in the high diversity context or low diversity context? And I thank you for the question because I'm saying that I actually should not have skipped that point because that's kind of like, you know, maybe 70% of why this is exciting. So, you know, thanks. And then we can, if I didn't address something, we can maybe discuss that separately.
Okay, we have time for one quick question.
Hey, thanks for that neat talk. I think sort of a similar question and problem set arises in genetics sometimes when you have a very complex phenotype that you can really only ascribe to sort of the epistatic interactions among hundreds or thousands of genes. And I'm sort of wondering if you're getting that like a type 3 error almost in some of those simple descriptions with a very few amount of parameters in your descriptive model. And if you could use like discriminating upon the function of some of these systems we're trying to do to get at if you're the smaller number of errors of the description complexity in your models can help you discriminate on if you're actually converging on a true process or if it's like a type 3 error and you're getting the right answer for the wrong reason. Does that make any sense?
So I always had trouble with type 1, type 2, type 3. It all depends on false positive, false negatives, and what's the type 3? Remind me?
If I can remember, I think it's like you're getting the right answer for the wrong reason.
Yeah. Yes. Okay. 2 comments on that, and for the rest, maybe take it offline because we can then discuss in more detail. 1 comment is this analogy to genetics that he just made is actually like spot on. For example, early on, it could be this whole thing with this part. What we did in this paper was precisely to get inspired by genetics, where, you know, what we do in ecology usually is I put some species, now they'll be interacting, and from the interactions, let me try to predict what abundance they will settle in, right? Which is a very complicated arrow here. And then given that, there's another very complicated arrow. What is the actual functional, like the functional property I'm interested in? How is it shaped by all these species now that I know their abundances, right? And in genetics, that's a perfect analog to something like I have this genotype and mutations that happen, they shape expression levels of all sorts of genes, which is very complicated, right? Let's try to predict that. That's a nightmare. But now imagine I've succeeded. Now I need to use that to predict, I don't know, fitness, and that's another nightmare. And geneticists for a long time have had massive success. So, you know, actually, given how simple this is, seems very surprising that let's just think about mutations. What is their effect on fitness, bypassing all of that, right? So the reason why it didn't seem entirely crazy to try something like this was precisely borrowing this landscape intuition from genetics. So totally, that's a very apt analogy. As for, are we getting right answer for the wrong reasons? So we try to make sure that doesn't happen by, that was our criteria for the data sets, to have enough data points for the predictors we had, so we could do randomization tests to make sure that we all, like, the prediction is out of sample and for the right reasons, right? So, you know, we do let's scramble the data, retrain the whole thing. Do I get this same behavior? If that's the case, then this is nonsense, right? And we don't. So we did those checks.
Okay, great. I think that wraps it up. We will have refreshments outside and you'll have time to talk to the speaker.